The royal skeletons looked powerful, right up to the moment their DNA showed the family folding inward. The first disturbing clue was not gold. It was not a sword, a crown, a carved seal, or a throne room. It was a pattern of matching DNA segments inside bodies that had been buried to look untouchable. The chamber had been arranged in the old language of power: stone walls, sealed graves, polished objects. Bodies set where rank could still be seen, even after speech and memory were gone. For generations, archaeologists read those scenes from the outside. Imported beads meant wealth, weapons meant status, monumental stone meant authority. A body near the center of a palace burial looked like a ruler, or someone protected by rulers. Then, ancient DNA changed the evidence. From the dense petrous bone of the skull, and from teeth that held fragments of blood and ancestry, researchers could read what no grave goods could confess. They could compare one genome with another. They could measure how much DNA two buried people shared, and whether that sharing looked ordinary, dynastic, or dangerously close. The question became sharper than any inscription. Were these royal dead related because dynasties remember their ancestors, or because power had been deliberately bred inside the same family line? The grave looked like authority. The genome showed the machinery behind it. The caveat matters here because this subject is easy to distort. Royal incest, in this story, means biologically close reproduction detected through ancient genomes. It does not mean gossip about every royal family. It does not mean moral spectacle. It does not mean that rank, culture, intelligence, worth, or identity can be reduced to blood. Ancient DNA can reveal biological relatedness. It cannot tell us every private motive behind that relatedness. It cannot show consent by itself. It cannot show affection, fear, law, ritual pressure, or palace politics inside a single body. A genome is powerful evidence, but it is not a diary. The method itself is stark. Every child receives long stretches of DNA from each parent. In most families, those two sets are different enough that the genome carries a natural mix of variation. But when the parents are close relatives, the child can inherit matching copies of the same ancestral segments from both sides. Those matching stretches are called runs of homozygosity. The phrase sounds technical, but the image is simple. Imagine opening 2 volumes in the same family archive and finding whole copied pages in both books. A few repeated phrases can appear in any old community. Entire copied chapters point to close parents. That is why petrous bones became so important. The petrous bone is a hard, dense part of the skull near the inner ear. In ancient burials, where heat, water, soil, and time destroy most soft traces, that bone can preserve DNA better than almost any other part of the skeleton. Researchers drill a tiny sample. They sequence broken fragments. They compare the genome across thousands or millions of positions. Then they calculate kinship coefficients, which estimate how closely 2 people were related. This does not turn the dead into a scandal column. It turns a palace burial into a family record that can be tested. Once the rules are clear, the objects in the tomb lose their monopoly over meaning. A gold pin can say rank, a seal can say office, a carved blade can say ceremony, but a genome can say father, daughter, sibling, half-sibling, uncle, niece, dynasty, bottleneck. And once that language is readable, some royal graves stop looking like family trees. They look like loops. Picture a palace burial complex at the height of its power. The dead are not thrown into the ground, they are staged. Stone chambers are built near the seat of authority. Doors are sealed. Vessels are placed beside the bodies. Weapons, jewelry, imported shells, ivory, lapis, bronze, or painted pottery mark the dead as people whose lives were tied to command. In an older reading, those objects did most of the speaking. A man with a weapon near the central chamber might be a warrior. A woman with ornaments might be a consort, priestess, queen, or noble relative. Children placed near adults might be heirs, sacrifices, servants, or symbols of continuity. The burial announced hierarchy. It did not announce biology. Then the laboratory adds a second voice. A tooth root from one body is sampled. A petrous bone from another is drilled under clean conditions. The DNA fragments are sequenced, copied by machines, and compared across the burial group. The result can overturn the room. The first evidence layer is status. These are not random graves scattered through a village. The architecture, objects, and ceremonial placement show elite treatment. The burial says that some bodies mattered more than others, at least to the people who arranged the dead. The second evidence layer is kinship. When those elite genomes are compared, the bodies often stop looking like unrelated nobles. They begin to form a biological household. A woman in one chamber may share half her genome with a young man in another. A child placed near a decorated adult may be that adult's son or daughter, not a symbolic offering. Two men buried as separate branches of authority may be brothers, half-brothers, or father and son. Palace grave goods say rank. Shared DNA says family. That distinction matters because ancient courts often used objects to create a public story. A seal could present office. A weapon could present strength. A robe, crown, or ritual vessel could present legitimacy. DNA can identify the biological structure beneath the performance. The first model broke there. Elite burial did not always mean a collection of servants and rulers, or strangers gathered by ritual. In several ancient DNA studies of high-status graves, the dead formed close kin groups. The burial was not only a political scene, it was a biological map of power. But close kinship alone does not prove incest. Dynasties are supposed to be related. Royal houses pass titles through families. Kings have sons, queens have daughters. Cousins appear in palaces because the palace itself is a family machine. The disturbing turn appears when the relationships become too close for ordinary dynastic distance. Parent and child share about half their DNA. Full siblings also share about half, but the pattern looks different across the genome. Half-siblings, grandparents, aunts, uncles, nieces, and nephews leave their own statistical traces. Ancient DNA researchers do not rely on a single matching point. They study the length and arrangement of shared segments across the entire genome. That is how a palace burial can move from "these people were related" to "this family structure was unusually closed." The Newgrange finding in Ireland became one of the clearest public benchmarks for this kind of evidence. It was not a palace in the later royal sense, but it was an elite monument, one of the most imposing ritual structures in prehistoric Europe. In 2020, researchers published genome-wide results from Neolithic Ireland in Nature, including an adult male buried at Newgrange whose parents were first-degree relatives. That means sibling level, or parent-child level. The signal was not rumor. It was not mythology. It was written across the long matching blocks of his DNA. After that, older burials had to be read with a sharper eye. A royal chamber could still be a statement of descent, but if the genome shows repeated closeness, the family tree no longer branches cleanly. It begins to close on itself. A single close union is one kind of evidence. A dynastic bottleneck is another. A bottleneck happens when a population, or a family line, passes through a narrow gate. Many possible ancestors are excluded. A small number of people contribute more of the next generation than chance would normally allow. Over time, the gene pool narrows. In an isolated village, a bottleneck can happen because geography limits marriage choices. A mountain valley, a small island, or a community cut off by distance may carry the signs of repeated marriage within the group. The pattern can be cultural, practical, or accidental. In a royal family, the narrowing can become political. Power is not only held by armies and walls, it is held by inheritance rules, sacred stories, land claims, priestly approval, and fear of outside families. A marriage can move property. A child can unite rival claims. A daughter can bring another house into the succession. A son can make a throne vulnerable if his mother's relatives become too powerful. Close marriage can appear, in that logic, as a way to seal the system. If power is believed to flow through a sacred bloodline, marrying outside that line can feel like dilution to the court. If land must stay inside one house, outsiders become a threat. If a ruler claims divine or ritual status, a sibling, cousin, niece, or half-sibling may be treated as the only acceptable partner for producing heirs. The genome kept one more record. Repeated cousin marriage leaves one kind of signature. Repeated closer unions leave a heavier one. The difference is visible in the length and number of identical DNA segments. Older shared ancestry creates shorter fragments, broken down across generations. Recent close parents leave long stretches that have not had time to be chopped apart by recombination. Recombination is the shuffling that happens when DNA is passed from parent to child. Each generation breaks and rearranges inherited segments. That is why distant ancestry usually appears as small pieces. Close ancestry appears as large blocks. In a royal bottleneck, researchers may see both patterns at once. There can be many short shared segments from older isolation and very long runs of homozygosity from recent close unions. Together, they show a family system that is not merely old, it is compressed. History gives careful parallels. Egyptian royal sibling marriage is documented in certain dynastic periods, especially where divine kingship and closed royal descent carried political meaning. In the Inca world, royal sibling union was tied to sacred kingship and succession. In Hawaiian chiefly systems, high-ranking sibling unions could concentrate sacred rank. These examples cannot be casually pasted onto every ancient burial. Each culture had its own law, ritual, and politics. But they prove one important point. In some elite systems, close kin marriage was not treated as ordinary family failure. It was built into the logic of rule. Texts sometimes tell us this directly. Many burials do not. That is where ancient DNA becomes so disruptive. It can test social structures in places where the written record is silent, lost, flattering, or controlled by the court itself. Inscriptions often name titles. They praise fathers. They honor mothers. They record victories, offerings, and divine favor. They rarely admit that a dynasty narrowed itself to protect power. In some elite burials, the body is not only the ruler. The body is evidence of the political system that produced the ruler. A skull in a chamber, a tooth in a jaw, and a child beside a ceremonial object can show how inheritance was managed long after the palace language has vanished. The disturbing part is not that ancient people had kinship rules different from ours. The disturbing part is the pattern that appears when power treats reproduction as statecraft. The body becomes policy. The strongest single image comes from Newgrange. The monument stands in Ireland's Boyne Valley, built more than 5,000 years ago, centuries before Stonehenge reached its later form, and long before the pyramids of Egypt. Its passage and chamber were aligned with the winter solstice sunrise, when light enters the tomb for a brief moment in the dark part of the year. This was not an ordinary grave. It was an engineered monument of stone, earth, ritual, and astronomical precision. Whoever was placed inside was not treated as a common villager. The man from Newgrange was an adult male. His bones did not come with a written confession. No tablet named his parents. No inscription explained the rules of his house. For a long time, the monument itself carried the awe, and the human story inside it stayed partly silent. Then, in 2020, researchers including scientists from Trinity College Dublin and ancient DNA specialists connected with Harvard published genome-wide results from Neolithic Ireland in Nature. Among the samples, the Newgrange man stood apart. His genome carried the unmistakable mark of first-degree incest. First degree means the parents were as close as siblings, or as close as parent and child. In genetic terms, the long matching segments across his genome were too extensive to be explained by distant cousin marriage or old community isolation. They pointed to a union inside the closest circle of family. The evidence points in a narrower direction. Ordinary communities usually build barriers around such unions. They may explain those barriers through law, taboo, religion, custom, or practical knowledge. The details vary across human societies, but the avoidance itself is widespread. That is why the Newgrange genome shocked researchers. It suggested that the man did not come from an ordinary household that accidentally broke a rule. He may have belonged to a protected elite class where different rules applied. The body was inside one of Europe's most famous monuments. The DNA inside that body suggested a family structure so closed that his parents were from the same immediate line. The monument did not have to say king. The genome said that his birth was politically unusual. The public phrase royal incest can sound like a later palace scandal, something from medieval courts or Egyptian tomb paintings. Newgrange is older and stranger. It comes from a Neolithic world without surviving royal lists, where power may have been expressed through monuments, ritual knowledge, labor control, and sacred descent rather than written crowns. That makes the DNA more important, not less. If a society builds a monument aligned with the sun, places a man inside, and that man carries the genetic signature of first-degree incest, the burial begins to suggest a special class. Not just wealth, not just high status, a class that may have controlled marriage and descent in ways ordinary people did not. The second layer was stranger. The Newgrange study did not only identify one unusual genome, it also connected elite treatment, monumental burial, and biological exception. The man's body became a small opening into a wider social order, one where ancestry may have been used to set certain people apart. There is no need to invent a crown to understand the danger. When descent is treated as sacred, the family can become a locked room. The people inside it may be honored, protected, displayed, and constrained at the same time. A court record, if it had existed, might have called the bloodline holy. A monument might have called it eternal. The genome called it close. That is the disturbing revelation. Not one forbidden birth alone, not one body that makes modern viewers uncomfortable. The deeper implication is that a society powerful enough to build monumental tombs may also have built a reproductive boundary around its highest line. Gold can be placed in a grave after death. Architecture can be raised by workers. Ritual can be performed for the living audience. But DNA is carried into the tomb by birth. One body can expose the scandal. Several bodies can expose the system. To understand why, the genetic mechanism has to be read carefully. Researchers distinguish ordinary endogamy, cousin marriage, and first-degree incest by looking at identical segments across the genome. The important clues are how many segments there are and how long they remain. Endogamy means marriage within a community or social group across many generations. It can create many shared ancestors in the background. In the genome, that often produces shorter matching segments because time has broken the old inheritance into smaller pieces. Cousin marriage creates longer segments than distant community endogamy because the shared ancestors are more recent. First-degree incest creates the longest and most severe pattern because the same immediate family line is feeding both sides of the child's genome. This is why runs of homozygosity are so powerful. They do not merely say two people shared an ancestor somewhere in the past. Their length can point to how close that ancestry was. A palace burial with one close relationship might show a personal event. A palace burial where several elite bodies show high relatedness, repeated long matching segments, and narrow descent begins to look different. It becomes a genetic record of succession strategy. At that point, the cemetery is no longer only a place where rulers were buried. It is a diagram of how power reproduced itself. The Y chromosome and mitochondrial DNA add another layer. The Y chromosome is usually passed from father to son. Mitochondrial DNA is passed from mothers to all their children. Together, they can show whether a ruling line was narrow on the male side, the female side, or both. A burial might show one dominant paternal line through the Y chromosome, suggesting that authority moved through men of the same house. At the same time, mitochondrial DNA might reveal women brought in from outside groups, or women repeatedly drawn from the same restricted circle. The contrast can be revealing. A court might record only a man's title. The Y chromosome can show whether several buried men belong to the same paternal line. A woman might be labeled by archaeologists as a foreign bride because of her ornaments. Mitochondrial DNA can show whether she was biologically local, closely related, or connected to another burial group entirely. Children are even more revealing. A child's genome can link two adults across separate graves. A young body in a side chamber may show that two elite branches were not separate after all. A supposed servant may turn out to be a relative. A supposed relative may turn out to be unrelated, placed there by status, ritual, or death practice, rather than blood. The genome can rebuild the hidden household. Half-siblings may be buried as if they were separate branches. A woman thought to be an outsider may be central to the line. A child can connect two chambers that architecture kept apart. Retainers can be shown not to be kin, even when their bodies were arranged close to rulers. This is where ancient DNA challenges the palace story most directly. Written records often preserve the names that power wanted remembered: father of the king, wife of the ruler, son of the divine house, daughter of the great line. Those titles may be true, but they are selective. DNA records the relationships behind the titles. It can reveal a father whose name was omitted. It can show that two people called cousins were actually half-siblings. It can show that a dynasty brought in outsiders at one generation and closed around itself in the next. It can show that an elite cemetery was not a broad noble class, but one narrow family repeating itself. That is where the old map failed. A family tree is supposed to spread. Parents produce children. Children marry outward. New lines join. Branches multiply. In a closed royal system, the branches bend back. The same ancestors appear on both sides. The same segments return. The genome grows less like a tree and more like a corridor with mirrors. The title's promise is disturbing because the evidence is not symbolic. It is measurable. Long matching blocks do not care about royal ceremony. Kinship coefficients do not flatter a dynasty. A petrous bone does not repeat court poetry. It preserves the biological structure. Still, the limits matter. Ancient DNA can reveal biological incest, dynastic bottlenecks, hidden parentage, and elite endogamy. It can show that a person's parents were very close relatives. It can show that a burial group was tied by blood in ways the objects did not reveal. It can show that a family line narrowed across generations. It cannot automatically reveal consent. It cannot tell us whether a union was celebrated, forced, feared, resisted, or required by law. It cannot tell us whether the people inside the system experienced it as sacred duty, political necessity, private harm, or something no modern category fully captures. The evidence can be disturbing without becoming a spectacle of shame. It is also important to be careful with medical claims. Reduced genetic diversity can raise the chance that harmful inherited variants appear in children, especially when close unions repeat. That is, the biological risk of narrowing a family system too tightly. But an ancient genome from one burial is not a full medical diagnosis of a person's life, health, or fate. The responsible conclusion is structural. Royal power often tried to make bloodlines look pure, sacred, continuous, and unbroken. Architecture helped. Ritual helped. Gold helped. Written titles helped. A body placed in the right chamber could make descent look inevitable. The genome shows the cost of closing that system. A palace burial advertises continuity. DNA can reveal compression. The grave says the line was protected. The genome may show that protection became a trap, because the family had turned inward to keep power from escaping. That is why ancient DNA changes these cold cases. It does not merely add another detail to archaeology. It changes the kind of question we can ask. Instead of only asking who was honored, we can ask how the honored were produced. Instead of only reading rank, we can read relatedness. Instead of only seeing a dynasty, we can see the biological pressure beneath dynastic myth. The skeletons at the opening looked powerful because the burial was designed to make them powerful. The chamber, the objects, and the placement all spoke for the court. Their DNA spoke for the family structure. What ancient DNA revealed about royal incest is disturbing because it shows how dynasties could turn family into a political machine. The machine could be hidden behind ritual. It could be covered with architecture. It could be polished with gold. It could be protected by silence. But the bones kept another record. In a sealed chamber, a body may still carry the truth of its parents. In a tooth root, a child may still connect two branches the court wanted separated. In the petrous bone, long after titles have faded, Matching DNA segments can name a closed bloodline without using a single royal word. That is the power of the genome archive. It does not replace history. It presses against history where history was edited, flattering, or incomplete. Subscribe to the Genome Archive for careful genetic history, ancient DNA cold cases, and the parts of human ancestry that written records left out. No hierarchy, no purity myth, no spectacle of shame. Only evidence, power, inheritance, and the human cost of treating blood as a throne.